Okay, thanks for holding on or holding out till this last session of today's talks. Today I want to present some work we've been doing on developmental plasticity and phenotypic, developmental plasticity and phenotypic divergence across different species of Spadefoot Toads. And this is work that Ivan and I have been doing for the last couple of years. So just to make sure we're all on the same page, phenotypic plasticity or phenotypic or developmental plasticity refers to the ability of an organism to change its phenotype or development in response to an environmental stimulus. And sort of two famous examples are the development of these spines in Daphnia in the presence of a predator, or the maintenance of the larval form even throughout sort of the reproductive phases of these pedomorphic salamanders. So these are widespread, and I'm sure we can all think of many examples of phenotypic plasticity, and they've been acknowledged throughout most of sort of the history of biology. But how we view its role, the role of phenotypic plasticity in evolution, that has changed somewhat in the last decade or two, specifically in how it can be a mechanism to bring about evolutionary novelty, such as the diversification of these ornamental structures in dung beetles. And the proposed mechanism through which this is possible has been termed genetic accommodation. So I'm going to run you through what the general gist of genetic accommodation is, but first I thought I would point out that very often when we talk about phenotypic plasticity, we talk about reaction norms. And reaction norms are essentially so for example, for this individual here that's inhabiting a heterogeneous environment, the same genetic individual can express one phenotype in one environment and another phenotype, or sort of a gradient of these phenotypes in another environment. And the slope, so the difference between these two phenotypes, we refer to as the reaction norm. So back to genetic accommodation, the idea here is that ancestrally you have an individual that, or sort of a genotype that inhabits heterozygous environments and expresses sort of this phenotypic plasticity, and then divergent lineages start to occupy more homogeneous environments. So they shift either to the purple or to the green parts of this environmental spectrum, and over time they lose that plasticity. So you see that the reaction norm becomes shallower. And this canalization of the phenotype can then result in divergence in the phenotypes. And the reason why this is deemed to be controversial is because you have phenotypic change occurring before you have genetic change, which is sort of the classic view of the modern synthesis. So today I'm going to talk about Spadefoot Toads, and the trait that we're interested in is larval period and the acceleration of larval period in these frogs. So as most of you know, frogs have these terrestrial adult phases and aquatic larval phases. And the time the frogs spent as larvae is different between species, but also within species. So there's plasticity in this larval period. And for example, in Pelobades, this is dictated by the drying out of ponds. So as the tadpoles sense the water levels dropping, they can accelerate their larval period to sort of evade imminent death caused by the desiccation of these ponds. So Spadefoot Toads present us with this really interesting system for looking at potentials for genetic accommodation as a mechanism in evolution, because in this one single group of frogs, we actually cover the whole range, or sort of much of the range of larval periods and larval period plasticity in amphibians. So on the one end of the spectrum, you have Scaphiopus cauchi. So this is a Spadefoot toad found in northern Mexico and southern USA. They inhabit extremely ephemeral pools. So these are arid environment species. They lay their eggs in almost these sort of puddle-like pools. And accordingly, their larval period is extremely short. So they go from an egg being laid in the water to the tadpole undergoing metamorphosis and leaving that pond in about a week. So that's pretty drastic. On the other end of the spectrum, you have Pelobates cutripes, that's Sapoespuela, the western Spadefoot toad that's found throughout most of the Iberian Peninsula. And they again inhabit these ephemeral systems. We're all familiar with these from Doñana, but these tend to be longer lasting ponds. Often they sort of exist for the entire season. And accordingly, their larval period is also substantially larger. So they have larval periods of usually about 3 to 6 months. Not only are they different, these two species, in their absolute larval period, but also in their plasticity in the larval period. So if we can take these tadpoles, we can rear them in standardized conditions, which we do here in the climatic chambers, and we can simulate pond drying by dropping the water level on a subset of these. And what we see in Pelobades is that they actually reduce the larval period in response to the stimulus. So they show a good 30% acceleration in their larval development. When you compare that to Scaphiopus cauchi, so we do the same experiment, we drop the water level, and we actually see sort of substantially less plasticity. So they just accelerate fast no matter what the environment. So a lot of work has been done on this, mostly by Ivan and his colleagues. And we've got a pretty good understanding of what's actually going on. So for example, we know a lot about the hormones, that are governing this plasticity. And two key hormones here are the thyroid hormone and corticosterone. And in response to the simulated pond drying, we see an increase in the endogenous levels of both of these. And when we look at Scaphiopus, again, we see sort of this canalized high expression of, or sort of high levels of these hormones. Similarly, we also know a lot about the physiology and the physiological consequences of sort of this environmental treatment. So for example, in Pelobades, we see an increase in the metabolic rates in response to pond drying. They use up all the fat reserves that they have, and they also metamorphose as smaller individuals as a response to this accelerated development. When I overlay the same reaction norms for Scaphiopus, you can see that again, sort of across the board, there's this canalization, so they show substantially less plasticity. And what's interesting is that they almost seem to represent sort of a canalized form of the accelerated morphotype or phenotype, of Pelobades in all of these cases. So to summarize, larval period acceleration is plastic in response to pond drying in Spadefoot toads, more so in one species than the other. So there's this clear divergence in reaction norms at species level. And but we're also seeing this plasticity within within one species, and sort of the plastic response we see in Pelobades seems to be mirroring what we're seeing between the two species as well. So we think these might be a good candidates for genetic accommodation. And what we wanted to do is to take a comparative transcriptomic approach to see whether we can see what's actually going on at the level of gene expression. And our aims here were to characterize the environmentally sensitive gene networks that are underlying this developmental acceleration. And we wanted to see how much of that is actually conserved across the two species and how their expression patterns differ. And we did have some hypothesis going in, so we thought that surely the plastic species we'd see a lot more differential gene expression because they're clearly more sensitive to the environmental stimuli. So to test this, we set up an Rna-Seq experiment. Basically what we did, we reared the tadpoles of both species under standardized conditions, and at the time when they're most, or at their developmental stage when they're most sensitive to this pond drying, we exposed a subset of these tadpoles to sort of reduced water levels for either 24, 48, or 72 hours. We didn't have a 72-hour category for Scaphiopus because if you remember, these guys develop extremely fast. And we had 3 biological replicates for each of these treatments and control, which is kind of the minimum you need to really be able to say anything statistically from the Rna-Seq data. So this next step is what we did in LEM. So we extracted total RNA from the whole bodies of these tadpoles, and then we sent off those samples to be sequenced on the Illumina platform, and we got about 30 million reads per sample. And then we did de novo transcriptome assembly using Trinity, and we annotated that with the complementary Trinotate software. And then all the differential gene expression, for those of you interested, were done using Callisto and EdgeR, and the geoterm assignment, which I'll talk about a little later, was done with GeoSeq. So this is what this looks like, and I guess most of you aren't familiar, so I thought I'd spend a little bit of time explaining that. So this is a heat map of the differentially expressed Trinity genes for Pelobades. So this is the plastic species, and what you see here is the treatments, so each individual per treatment along the x-axis here, and the y-axis, so the rows of this heat map represent differentially expressed genes. So these are genes or fragments of genes that show differential expression, so they're up or down regulated for at least one pairwise comparison for these treatments, with sort of the darker purple colors showing upregulation and greener colors showing downregulation. So for example, here you have like this big chunk of genes that basically has fairly sort of, central values of expression for the first 48 hours, and then experience a spike in expression in this last 72 hours. And kind of you see the reverse down here. I mean, it's a little messy, but basically you see a lot of genes that are being upregulated in response to pond drying, sort of, or sort of in those first 48 hours, and then sort of a downregulation of these genes after 72 hours. So we can plot the number, just looking at the number of differentially expressed genes, we can plot those sort of as pairwise comparisons to the control. And the first takeaway I want you to have from this is that, interestingly enough, sort of the immediate response to this environmental stimulus, to pond drying, is actually a down regulation of genes. So essentially these tadpoles are switching off a lot of their normal functions in response to this stimulus. And then sort of at 72 hours we see an inversion where then there's actually more upregulated genes than downregulated genes. So we spoke earlier about reaction norms and phenotypes, and we can do more or less the same thing with the expression of genes. So for example, here I've singled out one gene, retinoid X receptor beta, and this is known to be very sensitive to thyroid hormone levels. So we expected this to show us a variable expression pattern. And what you can see is that you have an increase in the counts per million. So there's an increase in this expression with time of exposure to this treatment. And what we want to do is we want to compare that to all the other genes. So the first step is to normalize that, so that across the different treatments, each gene has a mean expression of 0 with a standard deviation of 1. And then we just do that for all of them, so for all 1,200 differentially expressed genes. And then the next step is basically to pull them out. So we use this R package called mFuzz to create these clusters of genes that have the same reaction norms or similar reaction norms. So what we're left is with groups of genes that are behaving in a similar way when you expose them to this environmental stimulus. So here basically we found 5 clusters to be a good fit for all of these 1,000 something genes. And you can see that they each show sort of these different responses to pond drying. Cluster 3 was the one which had the most genes assigned to it. And obviously we're biologists. We want to know what does this actually mean in the real world. And one way we can do that is by assigning Go terms. So Maria spoke briefly about geoterms. So these are gene ontologies. And basically these are annotated lists of genes that perform a similar molecular, biological, or cellular function. And through performing these enrichment analyses, so these are basically just, it's a Fisher's exact test. It'll tell you whether the genes in the group that you're looking at, or sort of the functional terms that you're looking at, are more represented in your treatment versus your control, or in our case, in this cluster compared to the background, so compared to everything else that's differentially expressed. Usually you end up getting these networks because everything is sort of connected and you have genes that are associated to more than one of these geoterms. And interestingly, so this cluster 3, where we're seeing sort of this massive downregulation as an immediate response to pond desiccation, includes things like steroid biosynthesis processes or fatty acid and lipid metabolism, which we know from previous studies on model systems are highly sensitive to thyroid hormone levels. These other two clusters had similar percentages of genes assigned to them, and they basically show the opposite pattern. So this first cluster, you have an upregulation of genes after 48 hours, and in the second cluster, you have an upregulation after 72 hours. And this cluster up here had RNA polymerases and protein syntheses, GO terms associated to it, and this cluster 2, thyroid hormone metabolic processes and keratinization, which is interesting because keratinization only, it's most strongly associated to metamorphosis in amphibians. So at this stage, we're already seeing signs of, this accelerated development in the larva. So now we can switch over to the less plastic species, the Scaphiopus cauchi. So again, the same matrix I showed you earlier. And what I want you to focus on here is the insane number of differentially expressed genes. So we've got about 4 times the number of differentially expressed genes as in the plastic species. And curiously, sort of the matrix is also a lot cleaner. So this was the pairwise comparison for the plastic species, and when we overlap that with the supposedly non-plastic species, what we're seeing is sort of, we're talking about way more genes that are differentially expressed in response to this environmental stimulus, but the pattern more or less stays the same. So the first reaction is that we have way more downregulated genes than upregulated genes. So again, there seems to be a shutting off of a lot of functions. And then, as time of exposure increases, we're sort of moving towards a possible inversion. But again, we don't have this 72-hour category. So when we compare these functional enrichment terms, these gene ontology terms between the two species, we see that there's actually not that many terms that are overlapping. Those that are in both of these matrices are things like responses to stress and anatomical structural development. So I suppose those are fairly easy to interpret. Both species are experiencing this environmental stress and thereby accelerating their development. And then in the plastic species in Pelobades, we're seeing more cellular responses to hormones. So they are, or it seems like they are more sensitive to thyroid hormones, and the majority of these GO terms that were only enriched for Scaphiopus seem to be more related to sort of actual anatomical developments, so lung development and phototaxis, which again suggests that they're actually moving closer to metamorphosis, or much faster than Pelobades. So phototaxis, for example, is thought to be related to tadpoles moving towards shallower parts of the pond that they're inhabiting. We can also compare, we can also compare the GO terms across the different time periods. So not only sort of one species to the other. So this is a bit messy, but I'll try and break it down. So looking at Pelobades first. So these are the number of overlapping GO terms. And what I want you to look at is that actually there's very little overlap between the different time points. So at each time point for Pelobades, we're actually seeing sort of very marked differences in the functional development of the species. And if you compare that to Scaphiopus, there seems to be a lot of overlap. And then comparing those across, we see this asynchrony that we kind of expected already. So for example, the stuff that's being expressed after 24 hours of exposure in Scaphiopus seems to correspond most to the 72-hour of exposure in Pelobades. So we can see this strong asynchrony in the development. So I thought I'd leave it there for now. This is still an ongoing project. Sort of interesting takeaways is definitely that what we're seeing is sort of this, the majority of genes are downregulated in response to this environmental stimulus. Unexpectedly, the less plastic Scaphiopus seems to be more sensitive at the level of gene expression. And although there are some conserved functional categories, so there is an overlap in these geoterms, and we're also looking at differences in reaction norms, so I didn't get into that today. But there's potential sort of gene candidates that we want to look at for testing whether the mechanism of genetic accommodation could have played a role here. But many of the functional categories, actually most of the functional categories, are not conserved across the two species, and definitely not across the time points in the two species. So I want to thank you for listening, and thank you to all the LEM staff for always helping us out when we need you.